So this is a relatively modern discovery, which is actually very crucial in understanding what goes on. So the aquaporins, these are proteins that actually let water in at a faster rate than usual diffusion. Okay, so much like everything that has been taught to you so far, medical education is like drinking out of a fire hydrant. So this lecture is meant to be sipped slowly, like your tears. No? So when we're talking about aquaporins, meaning these are actively pushed in water channels. There are two of note, which I'll be showing you today, introducing to you today. The aquaporin 1, which is what we see in the choroid plexus. So as you can deduce, it is a part of the CSF secretion, the cerebrospinal fluid production. So this is something new, aquaporin 1. So usually seen only here in where you see those brush borders in your choroid plexus ependymal cells. The remainder of the circles that you see here are not aquaporin 1, but they are aquaporin 4. So to the neurology residents for attending today, this is what is relevant to us when we're talking about demyelinating disorders. So AQP4 is found almost everywhere else, in the basolateral membrane of the ependymal cells, here in the astrocyte feet, in the blood-brain barrier. So that gives you a clue how important it is. And as well as in the areas without blood-brain barrier, the so-called circumventricular organs, meaning it has a lot to do with water. Okay, where do we see it in the astrocytes? They're here found in the astrocytic feet, the end feet of the astrocytes. It tells you the importance of the astrocytes in terms of the water dynamics in the brain and in the blood-brain barrier. Okay, so this is just a picture of a normal brain, a normal astrocyte with normal aquaporin 4 channels. And here is when those aquaporin 4 channels are disordered, meaning they are not normal anymore. So this case is what we see in Alzheimer's disease, which may be a reason why that waste is not allowed to be cleared from Alzheimer's disease brains. So if the trash is not collected by the basurero, what's going to happen to the neighborhood? It's going to stink and people will die, if the waste is toxic. Which leads us to the last part of this new lecture, the so-called lymphatics. So we are traditionally taught in medical school and in neurology residency training that there are no lymphatics in the brain. To repeat, there are no lymphatics in Basing Se. So no lymphatics, but that is farthest from the truth. The truth was being hidden from us. We call it the lymphatics. So what is the lymphatics? So now, because of the new definitions of what the blood-brain barrier is, we start first with the essence of the blood-brain barrier, which is your neurovascular unit. So your neurovascular unit contains, as mentioned, your endothelial cell, your pericyte, and the astrocyte and feet. So that's your neurovascular unit because of the involvement here of the astrocyte as well as the neurons. The intact blood-brain barrier must have intact tight junctions. So if you zoom in on that part with the tight junction, this is, as I showed you earlier, things that are not usually transported must be actively transported using energy, hence the mitochondria here. Okay, so a bit of water may pass in passively, but a lot of water may be actively pushed in by the aquaporin 4 channels. Okay, so this is just a busy slide to show you where the neurovascular unit and the blood-brain barrier unit fits into the overall scheme of the lymphatics. So zooming in on that lymphatics, now to try to understand this system, we just cut through it one bit by bit, so we understand that. We simplify the picture first. First, the meningeal. The meninges have been lectured already by Dr. Batecalon, but I'm not sure if he mentioned that there are lymphatics there with the blood vessels that supply the meninges. That's your subarachnoid space up there. Dr. Gap Legaspi will lecture to you on the cerebrovascular anatomy, but it will be pushed to a later date, depending on when he's available. So around those blood vessels, the arteries are a space. This is called the Virchow-Robin space. Virchow as in VIRCHOW, ROBIN space. Though these are the paravascular spaces, this will be important when we're trying to understand what lymphatics means. So that's the vein here. So what happens is CSF enters that paravascular space. So around your arteries, around your veins, and into the brain parenchyma, the brain tissue. So diffusion pushes this. So the CSF mixes with the fluid around the arteries and the veins and enters your interstitial space. So that's ISF, interstitial fluid. These are the astrocytes, as I mentioned. So they form your blood-brain barrier, and they're there to help monitor what goes on around as well the perivascular spaces. And that's the neuron here. That's the astrocyte end foot. And you have the capillaries. And ideally, the waste products are cleared and drained into these perivascular spaces as well as the CSF. And these eventually end up in your cervical lymph nodes. So this is just another picture depicting the artery to vein flow of that CSF around the vessel into the surrounding brain tissue and then out again into the space around the veins, carrying with it all the waste products. Okay, and this undercuts what Dr. Batecalon will lecture, but not all of the CSF will drain into the arachnoid granulations up there, but they can also drain into the those holes where your cranial nerve foramina pass through. Okay. So what happens when you have brain damage inside the brain? A damaged brain means a damage to the blood vessels supplying the brain. So a damage to the blood-brain barrier, meaning there's leakage. So there's impaired flow of the CSF when you have a swollen brain. If impaired influx into that perivascular space, impaired exit or efflux. Then because of the leakage, those tight junctions are open. Your immune cells can penetrate the blood-brain barrier and go into the brain. Your damaged neuron attracts your microglial cells and attracts your pro-inflammatory substances. And once they find the damaged neurons, they will eat them and phagocytose them, and the cells will die. And those waste products will actually spread to the other cells and cause further poisoning of the other surrounding cells. So the waste product accumulates and your brain dies slowly. And as well, you have impaired drainage. This contributes to the waste accumulation inside your brain. So essentially, it's a basurero problem. Outside, and this is already beyond, I'm just mentioning it here, just to end the lecture. The involvement of the nervous system, which transcends even the gastrointestinal tract and the respiratory system. Okay, so that's it. I hope I overwhelmed you enough to open your eyes that there are more knowledge now that can help you with your future patients and will help us treat our patients with neurological disorders. So it is 3 o'clock and I'll check the questions if we have time.